I'm Dr. Jing Liu from Shantou University Medical College. Today, we will continue to discuss respiratory physiology. This is the last session of the respiratory physiology chapter. We have already talked about the whole process of breathing, lung volumes and capacity, ventilation and perfusion in the lungs, gas transport and exchange at the pulmonary and tissue levels. However, the breathing is not constant all the time. It will change under different conditions. When you finish a vigorous exercise, you may breathe very heavily, like this, trying to get as much oxygen as you can. Why could your body detect the needs of more oxygen? Today, we will discuss the regulation of breathing. After finishing this session, you are expected to be able to, first, describe the regulation of breathing by different respiratory centers. Then, understand the mechanism through which the central and peripheral chemoreceptors are stimulated. Last, explain the effect of blood, CO2, O2 concentration, and pH on ventilation. Respiratory centers are structures in the brain that produce and regulate breathing. To identify their locations, physiologists conduct cross-cut experiments in animal brainstem. If the cross-cut site was between the pons and midbrain, breathing was normal, indicating the respiratory center was below the midbrain. However, if the cross-cut was in the middle of pons, the breath became slow and deep, meaning that the upper part of pons was the pneumotaxic center, PC. Next, they cut between the pons and the medulla oblongata. Breathing was still present, but lost their rhythm, suggesting that the basic respiratory rhythm was controlled by the medulla oblongata. Finally, if the cross-cut was placed between the medulla oblongata and the spiral cord, respiration ceased. The above result confirms that the respiratory centers are located in the medulla oblongata and pons, divided into 3 collections of neurons based on their locations and functions. A dorsal respiratory group in the medulla controls inspiration and respiratory rhythm. The ventral one is responsible for expiration and enhanced inspiration during exercise. The upper one, PC, controls the respiratory rate and depth of breathing. The purpose of respiration is to maintain proper concentration of O2 and CO2, as well as pH in blood. The respiratory center is responsible for inspiration and expiration, but the intensity of breathing is changed according to your body's needs. The most important contributor here is chemical control that works through chemoreceptors to monitor the changes in blood, such as CO2, O2 concentration, and pH. Let's first discuss the central chemoreceptor, which is located on the surface of a medulla. The central chemoreceptor cannot be directly stimulated by the changes of CO2 or hydrogen ion concentration in the blood. Why? Because this area is highly sensitive to hydrogen ions in the cerebrospinal fluids, and the pH is tightly controlled. However, hydrogen ion cannot cross the blood-brain barrier, so has less effect on the central chemoreceptor compared with change in blood CO2, which have a potential indirect effect. CO2 can dissolve in water to form carbonic acid and release hydrogen ion to stimulate chemoreceptor directly to increase ventilation. Faster breathing gets rid of the excess CO2 and keeps arterial CO2 concentration within a normal range. How about O2? Changes of O2 concentration do not have any direct effect on the respiratory center. They act indirectly through the peripheral chemoreceptors. Peripheral chemoreceptors located in the carotid and aortic bodies play important roles in controlling respiration. The signals received by the carotid and aortic chemoreceptors are transmitted through the glossopharyngeal nerves and the vagus nerves, respectively. A low arterial O2 concentration will strongly stimulate the peripheral chemoreceptors to increase the alveolar ventilation. A raised CO2 concentration and a lowered pH will aid the effect of a low O2 in activating the peripheral chemoreceptors. As we just discussed, the changes of CO2 concentration can indirectly affect the medullary respiratory center. At the same time, CO2 also stimulates peripheral chemoreceptors to increase ventilation. A decreased pH in the blood promotes alveolar ventilation through stimulating peripheral chemoreceptors. Mild hypoxia will also activate the peripheral chemoreceptors, and the individuals will develop deep and fast respiration to get more oxygen. However, severe oxygen deficiency may cause damage to the respiratory center, so breathing is slowed down or may even stop. Let's go back to the vigorous exercise. In such conditions, O2 consumption and CO2 formation can increase 20 times. In healthy individuals, alveolar ventilation can increase accordingly, keeping arterial O2 CO2 concentration and pH close to normal. Here comes the summary. Today, we have discussed the respiratory centers and their components. I hope you can now explain the effect of blood, CO2, O2, and hydrogen ion concentrations on ventilation, regulated by the central and peripheral chemoreceptors. Here are the references. That's all for respiratory physiology. Thank you for joining us.